Hi everyone. So today we're going to kind of switch gears. Within this semester, a main theme has been analyzing DNA sequence variation, and now what we're going to switch over to is looking at how genes are expressed and how we can tell differences between specific genes being expressed for any particular group. And so, remember way back to when we were first defining bioinformatics in our first day of lecture. Bioinformatics is really this exploration of the analysis of layers of biological information with these omics approaches. And we've been focusing on genomics that you can see circled here, so you will recognize a lot of the things that we've covered in this semester. GWAS, whole exome sequencing, for example. And now we're going to hop right over here to transcriptomics. So this is looking at the entire transcript complement of a particular cell. And this includes technology like gene expression microarrays and RNA sequencing, commonly called RNA-seq. And basically, what we're now looking at is we've been really interested in this direct effect of a genotype on a particular phenotype of interest. So say that's disease susceptibility or whatever we happen to be interested in. But now we're looking at these indirect effects that can often happen. Involving, these SNPs can often cause changes in how these genes are expressed within any cellular environment, and in so doing, actually affect a phenotype of interest. As with the case that we saw in DNA sequencing technologies, our technology to evaluate and look at transcripts has also improved from the mid-1990s, when we were first able to look at a small number, the expression of a small number of genes using spotted cDNA being complementary DNA, which we'll go over a bit later. 2002, this kind of graduated to a whole genome expression profiling, and now in the modern day, we usually do RNA-seq, and that's using that next generation sequencing technology to allow us to look at the gene expression on a transcriptome-wide scale. And so just to define RNA sequencing, it is the use of next generation sequencing. So remember our Illumina instrument that produces all of our FASTQs that we then transition into all the other file types that we've been looking at this semester. So it's the same instrument generating the same basic data, right? Those short sequencing reads. But instead of using it to detect specific sequence variation between, say, our human reference and a particular sample, now what we're looking at is we're using those short reads to evaluate gene expression. And the transcriptome, just to define it, is the set of transcripts expressed at a given time by a cell or tissue. This includes all forms of RNA, and so going through your biology classes, I'm sure you've heard of tRNA, rRNA, microRNAs, non-coding RNAs. The one that's usually analyzed is mRNA. And the reason that mRNA is usually targeted for these types of studies is that, as we all know from our central dogma of molecular biology, the mRNA molecules are what are immediately translated into proteins. RNA sequencing is the most current method for the evaluation of gene expression on a transcriptome-wide scale. As we discussed previously with our evolution of the technology, hybridization methods such as microarray were most often used. And we won't spend a tremendous amount of time on this, just to pinpoint a few key differences between, how our technology has evolved and how RNA-seq is really sort of helping us, evaluate these transcripts on a transcriptome-wide scale. And so you see our resolution has increased from our tiling microarray. High throughput has certainly increased, lending itself to the next generation of sequencing instrument. Low background noise. We can do a number of things analytically as far as the application. And we've been able to allay some practical issues as well. So we're not going to focus on experimental design because we are focused on actually analyzing the specific type of data. But just to quickly touch on it, when we do set forward with this RNA-seq based study, we have an overall question of will the data generated answer the biological question of interest? And this involves, considerations like how we're going to construct our next generation sequencing library. We know that the transcriptome can vary based on, the time that we collect a particular sample or the tissue that we get that sample from. What sequencing depth do we want to use? So remember, we ask this question when we are evaluating, trying to identify a disease variant, for example. How much depth do we need so we're sure that when we see a change that's present in our short sequencing reads, how many do we need to be sure of that call? And the same kind of question comes here, how many transcript counts do we need to be sure that if we say, this particular gene is upregulated, that it actually is? And then, of course, with any statistical measure, you need to talk about number of replicates. That gives you, a valid statistical test. And also, it's important to mention the mRNA actually, comprises a vanishingly small percentage of the total RNA in a given cell. And so a lot of times when we are generating these libraries, we actually have a polyase selection step or a ribodepletion step to maximize the mRNA yield from a particular sample. And so this might look very familiar to you from, what we've been doing as far as evaluating short sequencing reads. As far as disease variant classification and analysis. But, very briefly, long RNAs that we see here at the top, our mRNA molecule, are first converted into a library of complementary DNA fragments. And so basically we use the enzyme reverse transcriptase to produce, to actually reverse transcribe from that mRNA back to DNA. And then after that cDNA is produced, we have a library of cDNA molecules. It actually, moves forward very similarly to a next-gen sequencing experiment where, we put on particular adapters that allow us to sequence them, generate millions to billions of these short sequencing reads off of these smaller pieces. And basically what we get is, you'll also recognize this particular file. We get fast queues, just like we would get if we were sequencing the DNA of an individual with a particular disease or trying to do something like that. the file type that we get bioinformatically is the same. Now what we do with the data afterwards becomes quite different. But to this point, we've simply performed a next generation sequencing experiment and gotten a FASTQ. So we have our FASTQ from our Illumina instrument. And now what we do, and so similarly to our next-gen sequencing, disease identification type stuff with whole exome sequencing, we take those short reads and we align them to our reference. And so something very similar in both, in kind of both of these avenues of exploring this biological data. So we have RNA-seq reads and possibly a reference sequence. A lot of times we will have a reference sequence, especially in this class where we're very human-centric. And our deliverable is we want to, either reconstruct full-length transcript sequences from short reads or align them to our reference genome and actually count those transcripts that we've sequenced. And then our second step is some sort of quantification of those sequencing reads to tell us if we can identify what's called differential expression. And this is if a gene is up or down regulated, compared to, any particular group, the groups that you are investigating in your experiment.